let's go on. So here we can get the first kind of utilization that we can get is hybridization. Hybridization means the attachment of a nucleic acid probe with an existing gene of our interest. Suppose this green colored single stranded DNA is gene of our interest, and we design this red colored probe which is complementary to this gene. So designing this complementary probe will allow us to attach or make a hybrid structure with this desired gene. So this is called the hybridization. The hybridization that we are talking about can be of nucleic acid hybridization, or we can also utilize different type of blotting techniques like southern blotting and northern blotting for DNA and RNA, and also in case of western blotting we can look for proteins, right? Now in this case of this kind of probes that we are seeing here, the probes can be designed with radio labeled molecules, radio labeled things like the phosphate for example, which are radio labeled. So this radio labeled probe can allow us to detect the position of our desired gene in a versatility or variety of different clones. Okay, now let us first discuss about in this video we will be talking about several sets of screening techniques. Now the first one is the screening of libraries by colony hybridization. Colony hybridization is a technique of screening desired gene out. Suppose this is a plate having different colonies and we need to take out our desired gene. Okay, now how can we do this? First of all, in any kind of this hybridization techniques and screening techniques, we must take our colony imprints onto a nylon paper. Because these nylon papers are easier to handle, we cannot handle this master plates because master plates are rare. We need to store them all the time. So we just take a nylon paper and make an imprint onto it, which will get the exact kind of replica of this master plate. So it is also called a replica uh, paper of nylon paper. Now if we put this replica onto another petri plate uh, containing media, it will be called a replica plating. We'll be seeing the importance of replica plating later, and we can also utilize replica plating for the screening. But in this case what we're doing, we're just transferring the imprints onto this nylon filter paper. After transferring the cells onto the filter paper, what we are doing, we take what we are doing, we just use some of the chemical treatment to this nylon paper. We use alkali. Now what alkali does is alkali will break and lyse the bacterial cell, and then it will denature the DNA. So use of alkali is very very important. Usually sodium hydroxide is used. It will lyse the bacteria and denature the DNA. So now we are getting a single stranded DNA sticking out of this nylon filter. Then what we do, we will add the solution containing the radioactive probe which is destined to bind against our desired DNA. Now these are the radioactive probes here. Now we'll add this. After adding this radioactive probe, this will bind to the exact desired sequence which is given here in this white colored region. So this is the desired sequence, and this small red colored radioactive probe will bind to it. So what we get, proper binding is done. After that we go through several washing steps which will wash away the unbound parts, and also we need to fix this attachment via UV cross-linking. In a very here also I forgot to mention it, UV cross-linking is required for fixing these cells onto the nylon membrane. Then we go through this probe binding and several washing or to remove the unwanted probe. Then we expose this nylon filter to the X-ray to develop the autoradiogram film, right? So now we get the autoradiogram, and in the autoradiogram we get the position of those probes which binds with the desired gene. So from by looking at the position we can tell which part of this nylon filter is containing our gene. Now we know now we get an imprint of from the master plate. So comparing this with our master plate, we can tell the desired gene is the location of the exact desired gene. For example, here this and this bacterial cell is containing our desired gene. We can tell it by comparing this X-ray autoradiogram with our master plate design. Okay, so that is the end result. Then we can take out these samples, take out these cells colonies, and we'll culture them, and we'll extract the DNA from it. Then we'll amplify the DNA via PCR, and we can do the further analysis with this. Again the process is illustrated in this picture also. So say this is the plate, so we transfer it to the nitrous filter, then we denature it with NaOH. Then we get attach this radio label probe which is P32 label probe here. After that we expose it to the X-ray, and then we get the presence of this region. So if we compare these two images of the autoradiogram and with the master plate, we can get this is a region, and also this region and this region. These three parts in the original plate is responsible for our desired gene. So we can tell that these three colonies of bacterial cell is containing our desired gene. So we can extract the genes from these colonies and can do further study.